All right. So what we're going to do today is go through what you could call two sort of separate topics, but I've kind of linked them together here, which is the biological topic is really extremophiles, organisms that live in extreme environments. But we're going to use extremophiles as sort of a model for talking about how people study microbes. And then that's going to lead into the next lecture, which is on microbes that are associated with people. So human-associated microbes, both the good and the bad and the ugly. And we're having another review session today right after class at 2 Wellman. If you want to come to that and ask questions, please do. So I wanted to just cover one thing from the previous lectures because I sort of mentioned this, but I didn't sort of really reiterate this issue. There were a couple questions about this, and I thought I would just cover it again. So we've talked about lateral gene transfer, which there are lots of details underlying it, but one of the key reasons we talk about it is that it leads to the creation of these chimeric organisms, organisms where different parts of their genome have different histories because there's DNA coming from one branch into another branch. So in the end, you can't actually draw the evolutionary history of this organism represented in green here by a single evolutionary tree because some of its genes came from its ancestor, its sort of parent here, and some genes got brought in from this other lineage by the lateral gene transfer process. And this is really important for a lot of aspects of biology and evolution, these sort of fact that organisms can have parts of their genomes with very distinct evolutionary histories. Now, on a much sort of shorter evolutionary timescale, the same thing happens with sexual recombination. So you have two parents from, usually somewhat distant parental lineages, hopefully, and you undergo a recombination where their DNA mixes, creating these new combinations. And so within species, especially within plants and animals and microbial eukaryotes where there is sexual recombination, this sexual recombination creates, in essence, what you could call chimeras. We don't really use that term for them because this is sort of mixing of DNA within a species. And lateral gene transfer can result in mixing of DNA between species or even across very large evolutionary distances. But the concept is very similar. You get DNA from different lineages coming together in one organism. And then there's a third process that creates this type of chimera, more like lateral gene transfer in many ways, which is the endosymbiosis. So you have an organism, it brings another entire organism inside of it. That now creates an organism where the host has one evolutionary history, the symbiont has another. Now it turns out, I sort of mentioned this before, but I'm going to mention it again, that on top of endosymbiosis, we have very frequently lateral gene transfer. So when organisms brought inside of them the proteobacterium that became the mitochondrion, over evolutionary time, a lot of the genes that were in the mitochondrial genome have moved to the nuclear genomes of their host. The same is true with chloroplast DNA and chloroplast genomes. So in the nuclear genomes of all eukaryotes, there are genes that their evolutionary history is from alpha proteobacteria, as well as other genes that their evolutionary history is sort of from the eukaryotic line, that is sort of sharing a sister relationship with archaea. And then in the Plantae lineage, we had another endosymbiosis that brought in the chloroplast, and some of those chloroplast genes moved to the nucleus of those organisms. So in organisms in the Plantae lineage, In the nuclear genome, there are genes that are the normal eukaryotic genes. There are the alpha proteobacterial genes that came from the mitochondrion, and there are the cyanobacterial genes that came from the chloroplast. When you have secondary endosymbioses, this continues to go on. So you can have these very complex patterns in some of the organism's genomes. And I sort of hinted at this before, but I just wanted to sort of re-explicitly cover this. So all of these create, in essence, chimeric organisms. Just this organism here, even if none of the DNA moves from the chloroplast or the mitochondrion into the nucleus, the different genomes here have very different evolutionary histories. So just that occurring on its own without any movement of the DNA also creates these chimeric lineages. Are there any questions about sort of chimeras and gene transfer? Okay, so what I want to do is this sort of mixing of topics here where we're going to talk about the biology of extremophiles a little bit and use it as an excuse to talk about how people have studied microorganisms, both sort of historically as well as today. So if you go to any environment, this is an overhead view of a hot spring in Yellowstone National Park. If you collect soil samples or water samples or skin samples, wherever you go, if you take those samples and look at them in the microscope, you're going to see microorganisms, basically, at least wherever you go on Earth. There are very few places on the planet where you're not going to see microbes in some of those samples. And somewhat surprisingly, people didn't really realize this for a long time. Even if you go to these extreme environments, this is true. So in one of these hot springs, sort of the central part of some of these hot springs is 95 degrees Celsius or something to that effect. There are microbes you can even find in those areas that are 95 degrees Celsius. If you go to these very salty environments where if you took a normal cell like E. coli bacterium and put it into this very salty environment, it will die very quickly. This is basically like concentrated soy sauce in some of these salty environments. And organisms are still able to live there and thrive there. If you go to the bottom of the ocean, there are volcanoes in the bottom of the ocean where there are cracks in the seafloor and there's hot water coming up that has been mixing with the magma below. Because of the pressure of the water, so this is, say, 2 miles down in the bottom of the ocean, because of the pressure from the layers of water above, the boiling point of water down here in these hydrothermal vents is higher than 100 degrees Celsius. The pressure makes the boiling point higher. And there are organisms that live in those conditions. So the cap on the temperature of life isn't even 100 degrees Celsius. It's basically the boiling point of water, and in places where the boiling point is higher, there are microbes that live in those environments. And I've showed this before when I talked about the functional diversity in microbes. Again, if you look in all these sort of the tail end of the distribution pattern places where organisms are found, like this is Mono Lake, and this is in Antarctica. This was a postdoc of mine was studying microbes on Mount Erebus, which is the southernmost active volcano on the planet in Antarctica. And wherever you go, again, you find microbes. You generally are less likely to find multicellular organisms in many of these places, and we'll talk in a little bit of detail about some of this. So you go to these places, you collect a sample, you look at them in the microscope, and you can see diversity like this. This is the diversity of form that I showed you earlier. And so you can use that to try and make sense out of some of the things in the environment. And in general, what people want to do when they study any environment and any types of organisms, we can reduce sort of the big picture questions that they're asking about the microbes in those environments to these two questions. What are they? That is, what are the kinds of microbes that are found in that place? Just like if you went to an island and you saw big green leafy things or feathery or furry things, you would want to know what they were. People want to know what kinds of organisms are found when you look in these microbial environments. Making a list of the kinds is useful for many purposes, but really what people want to know is what are those organisms doing in that environment? What are their functions? What are their metabolic properties? Who do they interact with, etc. And that's what we're going to talk about today, is really how people answer these types of questions about microbes. And again, we're going to focus on extremophiles, but this principle applies to any environment that you go to where microbes are found. And what we're going to talk about can apply to any type of microbe: bacteria, archaea, microbial eukaryotes, and even many of these things can apply to viruses. So we're going to talk about 3 methods that people use to study microbes when they're interested in microbes in a particular place. You can go to the environment and study the microbes directly in the environment in which they reside. You can take a sample back to the laboratory and grow some of the organisms from the laboratory from the environment back in the lab. And then the last thing, which we may get to at the end of today or at the beginning of the next lecture, is what you could call CSI microbiology, which is we can study the microbes in the environment simply by looking at their DNA, by collecting DNA from environmental samples and using a DNA as an analog of what's going on in the environment. So first we're going to talk about this sort of field observation, which is a routine and very important tool in all of biology, especially things like ecology, but you can go to the environment and study organisms. And for macroorganisms, plants and animals, this is of course very common. So this, I used to work on butterflies, by the way. That is not me. Anybody know who that is? No, good guess though. It's Vladimir Nabokov, the author, who also happened to be the curator of Lepidoptery at Harvard. He's a very famous butterfly expert, but also a famous writer. In his autobiography, there are these great stories about him going and driving along a highway in Colorado and pulling over to the side of the road with his butterfly net when he was like 60 years old and running into some farmer's field before the farmer started to chase him away. But you can go out and collect organisms, all sorts of different organisms, if you study plants and animals in particular, and learn something about them. You can go to a field site and do some sort of just systematic looking at the organisms in the field site. You can use satellite imagery to study the forests in the Amazon, for example. You can be more systematic about it and do things like what's called building a quadrat, where you set up a specific grid of particular sizes and look at the organisms in the different squares there and compare and contrast them to each other. You can do more close field observations than with the satellite. This is probably why I stopped doing bird studies. Poor kid. And you can do this in any type of environment. Right? So you can go to all sorts of different places and study the organisms that live there. There's not, any particular constraint, although it's harder the more remote and difficult the places are. And all of these same general approaches you can and people do use for microbes. So this is another hot spring from Yellowstone National Park that's very heavily studied, and you can go to that environment, and you wouldn't want to put your hand into this hot spring, but you can put a sampler into it and collect the organisms and study them right there in the field, just like you might do with butterflies. You can, collect the microbes in ocean water and study them as you sail around the world. We use satellite imagery to learn things about microbes, such as based upon chlorophyll absorbance. That's one of the pigments used for photosynthesis. You can do this systematic type of approach by doing these quadrats. You can look in a microscope, bring it with you, so that'll help you understand things. And again, you can do this in any type of environment, and in fact, people do. There are field microbiology studies everywhere. So for example, this postdoc in my lab was on Mount Erebus in Antarctica in minus 60 degrees weather and very unhappy most of the time, but you can do the field work in these places. So the first thing we're going to talk about is if you go to the field, how do you identify what microbes are there? Who is out there? The way you're going to do it basically is to look in the microscope. And you're going to try to do something like what you would do with plants and animals. So I don't know how many people go out and look at birds or butterflies or lizards or whatever and carry around a field guide. You have a guide that tells you what organisms look like and where you expect them to be found and how to identify them. We have some of those for microbes. In general, that approach, looking at things in a microscope, does not work very well for bacteria and archaea. And that's because the appearance of bacteria and archaea is not a good indicator of what type they are. There aren't a lot of features that you can look at. In essence, 75 different types of bacteria look like this, and 300 types look like this. And every once in a while there's some type of bacteria that has a somewhat unique phenotype, but they're still not focused on an individual lineage. For microbial eukaryotes, you can sometimes identify them very specifically with just microscopic observations. There are certain diatoms, certain dinoflagellates, certain apicomplexans, all those lineages that we talked about. There are some of them you can identify the organisms just directly from observations, although many of them that hasn't worked out so well. So for example, the Phytophthora, the causative agent of the potato leaf blight that people thought was a fungus for many years, is pretty poorly classified by appearance. But appearance does work a little bit better for microbial eukaryotes than it does for bacteria and archaea. So you would use this in the field to try and identify things with many caveats. You can do the same sort of general thing by trying to figure out what they're doing. So we can use satellite imagery to find and characterize microbes in a variety of ways. This is an image of some bioluminescent microbes in the Indian Ocean. If you know something about the wavelengths that different organisms absorb at, you can make better sense out of this satellite imagery data. So certain proteins or pigments in microbes absorb at different wavelengths, and we can use the satellite data to figure out something about the abundance of different organisms from that information. And you can do this on a small scale too, so you can go to a local pond and study the functional diversity present in those samples. Well, I must have left out a slide. So you can go study the functional diversity in those samples, and there are lots of things you can learn. You can study carbon fixation. You can study nitrogen movement in particular samples. You can do sort of big scale studies of a place. One of the big problems, though, is you still have this trouble connecting the function to particular organisms. And one of the reasons is what I've already sort of suggested before, but I just want to give you an example type of question like this. So, which of the following is a reason why morphological traits of bacteria and archaea might not be useful for determining the type of microbe? So I've told you that it's not useful. Which of these explanations is sort of a rephrasing of why it's not useful? Right, so C, there is a lot of homoplasia in these morphological traits. So if you have the bacillus form of bacteria and archaea, this elongated rod, we see lots of different lineages, in essence, inventing that shape separately in bacteria and archaea. And therefore, it's homoplasious and not a useful metric for building evolutionary trees. And this is a big problem. So it's not just that you can't identify what type. If you go to an environment and you measure some function, you may have no idea what organism is carrying out that function. And people sort of who work on microbes always say that if you go to an island and you know that there's photosynthesis on the island, it's useful to know that it's in the plants and not the squirrels. So the same thing is true for microbes. If you can't partition the functions into what organisms are doing that function, it can be challenging to interpret what's going on in an environment. There's another problem which has really nothing to do with the appearance of microbes, but it's that they're small. And if you want to study the function, say, carbon fixation in a sample, and you go look at a pond water sample, there may be 10,000 different microbes in that sample, each of them present in 10 to 1,000 cells. Those are very small things. Putting your carbon dioxide sensor probe into that sample and figuring out which organism is doing what is not always easy. There are methods that people have developed to start to do this on a single cell basis, but it's still very, very challenging to figure out what's going on inside cells. So for these and other reasons, field work in microbiology can be a little bit more challenging than field work for other organisms. And so much more what people have done for microbes to try and figure out what's going on in an environment is to do a surrogate, which is you go to the environment, you bring the samples back to the lab, and you grow the organisms from that environment back in the lab. And you use that to try and study their types, and their functions. So there's a general term that's used for this called culturing or cultivation. They're sort of used interchangeably. And that's the growth of a microorganism in defined conditions, controlled or defined conditions. So you make some sample, say in a test tube or on a plate or something like that, where you know what you're putting into it. And then you can try and grow organisms in that condition. That's called culturing. And people do this routinely for microbes from all sorts of different environments. There's a particular aspect of this that is even more useful, which is what's called getting a pure culture. So in many cases, you can go make a growth media for some particular environment, put a pond water sample on that, and it's still got hundreds to thousands of different things in it. It's still hard to interpret in the lab. It may have less types of organisms than you had in the field, but it still can be hard. So what people try to do is they basically try to dilute their sample until they get single cells from the environment isolated from each other. And then you try and grow those single cells, get them to replicate in your defined conditions, and you grow up a large collection of clones of those single cells. So you get the single cell to replicate by asexual reproduction usually, either binary fission for bacteria and archaea or mitosis for microbial eukaryotes. And then you grow that up in the lab and you study it. Yeah?
Will you have a sort of unrepresentative genotypes as it might just be one strain?
You mean the thing that you grow?
Yes.
Yeah, so that...
One ancestor.
That unfortunately, that's what happens when you grow these pure cultures. So for throughout history, what people have frequently tried to do is grow many different clones from an environment and study their differences and see if there's anything that they share in common to try and see if there's an effect of maybe the genotype. Because you're basically getting a single thing to grow and make copies of itself, and who knows if it's representative of the environment. As we'll come back to at the end of the lecture today, this is why many people are going and using DNA sampling in the environment, because we can sample everything. And even if we're not growing them, we can at least sample everything in a representative way compared to their abundance in the environmental sample. And that partially takes care of your question, although not completely. So if you think about what microbes need to live, we talked about the different forms of trophy, the different nutritional sort of classifications for microbes. That's one of the common things that people vary when they try and grow organisms from the environment. So we can give them different energy sources. We can give them different electron sources. We can give them different carbon sources. And then you can give all sorts of variation in every other environmental parameter you can imagine. pH, temperature, oxygen, salt, any of those other things. You can control that and then see what you can grow from your environmental sample with those different conditions. So people have probably done a little bit of microbiology in labs here and there. I'm just going to show you, sometimes you grow things in a test tube. Sometimes you grow them on these plates that have basically jello on them with some food inside the jello. It's called agar usually. And what is done here, what you see on this plate, is someone tried to dilute a particular sample. So you take a little sample out of your, say, pond water, and you drag it with a stick or a loop across your little plate here. And then you take a clean loop or stick, or you sterilize it, and you drag it across again. You do it again, you drag it across again, and you keep doing this until you're diluting the sample, such that in the end, you have single cells at each of these points here, and they grow up as a colony when they replicate. So they make lots of copies of themselves, and that's what ends up being a colony on one of these plates, is clones of a single cell. And that's getting a microbe into pure culture. So you can now take that colony, separate it out from everything else, and in essence it should be one lineage of microbe, one species, one type, whatever, compared to everything else. The way this works is, again, by asexual reproduction, so that one cell will divide on that plate by binary fission, again, if it's a bacteria and archaea, by mitosis, if it's a microbial eukaryote, make lots of copies until those colonies are billions of cells stacked together into one spot. Now, you can have mutations during the growth of your culture. That creates a little bit of a complication, but these cells that you get at the end are pretty genetically uniform, especially compared to what you would find in pond water. And you can do this to get cultures, pure cultures, of hundreds to thousands to tens of thousands of different things from a single sample. And that's really useful because it's a lot easier to do lots of different types of experiments on billions to trillions of cells that are identical to each other instead of on single cells. So you grow up this batch of those cells. You can make hundreds of liters of some of them if you really need it. And then you can do biochemistry and physiology and genetics and genomics and sequencing and all sorts of other things with that large volume of material. So now I'm going to take that assumption that you have something in pure culture. How are we going to now figure out what it's doing and what it is? Who is out there and what are they doing? So if we've grown it up in the laboratory with billions of cells, we can look at them just like we did in the environmental sample. Now we have maybe more material, so we might be able to better detect some of the phenotypic pointers that tell us what type of organism is there. But in general, just like in the environmental sample, for bacteria and archaea, that's not going to work very well. And for some microbial eukaryotes, that's not going to work very well. So what everybody basically does today, if you can, is to sequence ribosomal RNA genes from your cultures that you've grown up in the laboratory to figure out what they are. And this is how it works. You take your sample, you extract DNA from that sample. Hopefully it's basically uniform. You might extract RNA, so that's what Carl Woese did many years ago. He was looking at the RNA directly. But the ribosome, with the ribosomal RNA inside of it, that's what he was looking at. There are genes in the genome of all these organisms that code for those ribosomal RNAs, so you could look at the DNA or the ribosomal RNA. So you extract those nucleic acids, and now you want to enrich them for those that correspond to the ribosomal RNA. And the reason you want to do this is that you want to read the sequence of those pieces, the string of letters inside those genes. It's hard to do if you have lots of DNA from all the different genes in the genome of that organism. You want to enrich, basically make billions or millions or trillions of copies of the ribosomal RNA from that organism in order to do this reaction where we're going to read the string of letters. Carl Woese did this by just purifying ribosomes from cells. There are thousands of ribosomes in individual cells, and total from a big culture you can get lots of ribosomal RNA. Now what we do, and I'll tell you about this in a minute, is we get the DNA and then we make copies of the ribosomal RNA genes. And I'll tell you how that works in 1 second. But what you do is you then sequence, you read the string of letters inside those genes. It would look something like this. And you compare them to the ribosomal RNA genes from other organisms in a data matrix. You make an alignment. So you line up the corresponding regions of the ribosomal RNA from different organisms, just like we line up the bones from different organisms, say different vertebrates. As I mentioned before, you can use the 2- and 3-dimensional structure of the ribosomal RNA to guide your alignment, just like people would use the position of bones in a skeleton to guide the alignment of bones. But in the end, you get this data matrix, and you build a phylogenetic tree of that data matrix. And if you have a new organism, you can see where it sits in the phylogenetic tree compared to organisms that you already know something about. So in this case, the organism in my 4 taxa unrooted tree that you should be highly familiar with here, groups on this side of the tree. We haven't rooted the tree, but let's just say we knew the root was in the middle, so we could say that this thing is more closely related to E. coli than it is to yeast in humans. The way you really do this is with thousands of ribosomal RNA sequences, and you can tell what type of organism you're looking at. So you grow the organism, you purify its DNA, look at its ribosomal RNA genes, and then you build a tree and you classify it that way. That's basically what Carl Woese did with all sorts of different organisms that could be grown in the laboratory, read the ribosomal RNA genes, and built this tree. And again, we're going to do the same thing and figure out where our new organism sits relative to everything else. And that's how we're going to give it a name, basically. So what I was referring to before is this sort of variant on this activity, which is what we all do today, which is you grow the organism, you extract the DNA, and you run a special chemical reaction called PCR, also known as the polymerase chain reaction, which can specifically make DNA copies of the ribosomal RNA coding region of the genome and make billions of those copies in a chemical reaction in a few hours. And then we read the sequence of those for our organism and build the tree. So that makes it a lot easier than what Carl Woese did. And this is now the routine method of classifying microbes that you grow in the lab. So you can go to an environment like the human gut, pull out samples from that gut, grow the things that you can grow in the lab, type all the different things that you can grow by this ribosomal RNA method, and you can get a list of the organisms that you find in different places. So that's how we're going to get the who is out there from an environmental sample, even though we're not working in the environment. We've taken the sample back to the lab and grown the things and then listed what we were able to grow. Now that's not, completely sufficient. That's not all what you want to do. What you want to do is figure out what organisms are doing. And again, we can do the analogous thing in the laboratory. So we can take an environmental sample like one of these high salt environments, grow the organisms from that environment in the lab, get them into pure culture in isolation, and then this allows us to do all sorts of experiments where we can figure out their biology. So, for example, if you wanted to know, you go to an environment, you grow something from the environment. You just got it to grow. But you don't really know if it likes those conditions that you gave it more than any other condition. So what you need to do now is vary some of those things that you added to the growth media and see which of those, how you vary, say, the pH or the temperature or the oxygen concentration. In this case, we're going to vary the salt. And we're going to try and figure out what this organism's favorite salt concentration is. So you go to the environment, just because it's living in 4 molar salt does not mean that it actually prefers to grow in 4 molar salt. So in the lab, we can figure out what the basic preferences is for different organisms. And this is really important in studies of things like extremophiles, because things might just be tolerating an environmental condition, and if nothing else can tolerate it, you'll find them there, but that doesn't mean that they prefer those conditions. So the way you would study this is you'd go to your environmental sample, get your pure culture, grow up a batch of that pure culture. It's called a starter culture. Set up some flasks with media that the organism likes to grow, and now you vary the thing you want to test in those flasks, like salt concentration. You take a little sample from your starter culture and put it in each of these tubes. Add a little bit of a small portion. And let's just say we compare 1 molar, 2 molar, 3 molar, and 4 molar salt. And now we monitor these over time to look at how the organism is growing in these different conditions. And the way we do that is there are a lot of different ways to do this, but one way is you can measure in essence, the density of stuff in your culture. Many microorganisms, if you shine a light through the sample, they scatter the light. So the more microbes that are there, the more they scatter the light, and you can quantify how much is there by those types of scattering experiments. So you monitor them over time. Say after 1 hour it looks like this, after 2 hours it looks like that, after 3 hours it looks like that. We take that data and we plot it. So we plot the absorbance or the amount of material in those tubes over time for each of the different conditions. And from this, we can now calculate a growth rate, that is the amount of doubling of cells per unit time, and plot it like this. So now we have salt concentration versus the growth rate of the organism. We had to do multiple time points to calculate a growth rate, but that now tells us that of these 4 conditions, this organism grew the fastest in 3 molar salt. Of all these conditions, that's its preference in terms of growth rate. if you do this type of experiment from organisms from lots of different environments, we can classify them by their salt preferences. So, for example, most organisms are what you would call non-halophiles, so like E. coli. If you raise the salt concentrations in their growth conditions, they die. There are other organisms that you could call halotolerant. That is, they can tolerate slightly higher salt concentrations, but their growth rate starts going down and down and down as you add more and more salt. They still survive, but their optimal growth rate is at very low salt concentrations. There are others that are called halophiles that once you cross a particular threshold, I think it's probably like 1.5 molar salt concentration, we're going to call them halophiles. That is, they like salt if their optimal growth is in salt concentrations that's above, say, 1 or 1.5 molar. And there are many examples of these halophiles, but most organisms are not halophiles. What's really interesting is that there are some organisms that are what we're going to call extreme halophiles. They basically do not like to grow until you make the salt concentration almost to the point where the salt is precipitating out of the solution. So that's like 4 molar salt concentrations in some conditions. Some of them will not even grow at all at lower salt concentrations. So we're going to call those extreme halophiles. Many people have been interested in how these organisms are able to grow in these very high salt conditions. And the reason for this is if you take a normal cell and you put it in an environment where there is lots of salt, the water inside the cell is going to want to leave the cell in order to balance what's called the osmolarity in the conditions. The water is going to migrate to where there are all these things dissolved. That's what I've tried to draw here, things dissolved in the water. The water is going to leave the cell, and the cell will desiccate, dry up. That's what happens to E. coli if you put it in high salt concentrations. So how are organisms able to survive in conditions where they're surrounded by all this salt and their cells should be very unhappy? What people have found by studying different halophiles and extreme halophiles is that there are sort of classes of adaptations that most of them use. What all of them do is they increase the concentration of things dissolved inside the cell. Increase the osmolarity inside their cells. So they fill up their cells with things like sugar, carbohydrates, or proteins, so that the water doesn't try and leave their cell all the time. Some of them use salts inside their cell, which we'll come back to in a minute, but that's pretty rare. Many of them build pumps inside their membranes to keep pumping things around so that they can keep the water from completely leaving their cells. And many of them can't do this perfectly all the time, but they have evolved means to be very resistant to drying, to desiccation. And this is pretty common across organisms. One of these or all of these adaptations are seen in most halophiles. There's one that I want to tell you about that is particularly unusual, which is in one lineage, basically, in one monophyletic group of archaea, the organisms fill up the inside of their cell with salts, which is very unusual. And this is unusual in part because when you fill up the inside of your cell with salts, if you do this to a normal cell, most of the proteins and other components inside the cell will not work correctly. Those salts screw things up. But the extremely halophilic archaea, this one lineage of archaea, has figured out how to fill up the inside of their cell with salts. And that's how they balance the salt in their environment. This is really important for biotechnology because there are many reactions that industries would like to carry out where there's just a lot of salt around. And we like to purify enzymes from various organisms to use them in these industrial processes, like things for laundry detergents or things for biofuel generation. And if you want to find enzymes that are adapted to working in high salt concentrations, you should go to extreme halophiles because all of their enzymes inside the cell work with very high salt concentrations. So again, this is one monophyletic lineage within the Euryarchaeota, where all of the organisms in that group are extreme halophiles, and all of them use this, build up the salt inside the cell as an adaptation. So this general approach, study the organisms in different environmental conditions and look at how they're able to adapt to extreme conditions, we can do with temperature too. So you can go to organisms from different environments where the temperature is high or low or whatever. We can do the same type of experiment, try and grow some of them in the laboratory, grow the starter culture, set up flasks with the same media in these different starter cultures, and now we're going to vary the temperature in these conditions instead of the salt. So let's say we do 20, 30, 40, and 50 degrees. Monitor the growth over time, and look at the growth over time in these conditions. Plot a growth rate or a growth curve, convert that to a growth rate, and now we're calculating, instead of the optimal salt conditions, the optimal growth temperature for different organisms. So now we can look at the differences in optimal growth temperature across different organisms. We see, for example, there are some called Microphiles that they prefer to grow at below 10 degrees Celsius. Some of them even down to basically 1 degree Celsius. If there's liquid water there, they can grow. The ones like us are called Mesophiles. That's in the blue curve here. Those are the ones that grow at sort of medium temperatures, prefer to grow at those medium temperatures. When you cross over to about 60 degrees as your preferred growth temperature, those organisms are called Thermophiles, preferring high temperature. When you cross over about 85 degrees, those organisms are called hyperthermophiles. You can even have organisms that grow above 100 degrees, that prefer to grow above 100 degrees Celsius. So on the surface of the planet, water is boiling at that temperature. That will eventually kill everything. But in the bottom of the ocean, where the pressure from the water above compresses the water below, the boiling temperature of water is higher than 100 degrees Celsius. And there are organisms in the bottom of the ocean, where there are volcanoes in the bottom of the ocean, that their optimal growth temperature has been found to be up to 110 degrees Celsius. Nobody thought that this was possible prior to sort of going to these environments and trying to grow things from these environments. There was theory that said, oh, there's no way anything can survive above about 70 degrees Celsius. But we find lots of organisms in these environments. If you classify the organisms that are found to be thermophiles or hyperthermophiles, we see that there are very few eukaryotes, even the microbial eukaryotes, that are able to grow at those high temperatures. There are many bacteria that are able to grow that are thermophiles. And there are some bacteria that are hyperthermophiles, but most hyperthermophiles are archaea. And once you cross over about 90 degrees Celsius, the only thing you see are archaea. So basically, the highest growth temperature that we see for archaea is the highest of organisms. And if you go to environments that the temperature in those environments is something like 95 degrees Celsius, you'll really only see archaea in many of those environments. Bacteria are able to grow at high temperatures, but not quite as high as archaea. And eukaryotes, kind of lame in terms of temperature growth. If you map the ability to grow at high temperatures, that is thermophiles, onto the tree of life, we see patterns like this, and this is an underrepresentation. This is the sort of full tree of bacteria and the full tree of archaea. You see thermophiles all throughout the bacterial portion of the tree and all throughout the archaeal portion of the tree. What appears to have gone on here is multiple separate inventions of the ability to grow at high temperature. Even hyperthermophiles seem to have come up many times. So this is a homoplasious trait across bacteria and archaea. Whereas extreme halophily and the use of salt as that compatible solute, that's only seen in one lineage of archaea. So this is contrasting different sort of extremophiles, different conditions. There are different sort of patterns in terms of the evolutionary mapping of these onto the tree. Yeah?
On the sample midterm, is asked like which one is studying for thermophile. Like, so in this case, it would be both of them.
So in that sample midterm, the question was if the temperature was 95 degrees Celsius, and basically we really only see archaea at those temperatures. So, and you know, the eukaryotes drop out basically at like 60 degrees Celsius. The bacteria drop out around 90 or 95, and then the archaea dominate once you cross over. So just like with halophily, we can look at the sort of the expectation of what the problem is at growing at high temperatures. So for mesophiles, if you put them in high temperatures, you would expect that their proteins are going to unfold. It's called denaturing. The RNA and DNA should unfold too. Chemical reactions for all enzymes should speed up. They increase with higher temperature. And the membranes of cells should basically liquefy. It's going to be too fluid, their membranes. And we see in thermophiles adaptations to deal with each of these issues. So to deal with the instability of proteins, thermophiles modify the amino acid content of their proteins. So they could use the same proteins, say a ribosomal protein or an enzyme that copies DNA. But they modify the amino acid composition of those proteins such that they're stuck together more at low temperatures. At high temperatures, they basically seem to have the same general structure as the same protein from a mesophile would at low temperatures. So basically, organisms are playing with the composition of their proteins in order to make them have the right stability at different temperatures. At high temperatures, most enzymatic reactions would, if you took E. coli and put them in high temperatures, all of the enzymatic processes are going to go crazy. So thermophiles tend to, in essence, dumb down their enzymes to slow down their reactions. If they were in low temperatures, they would basically not work at all. But at high temperatures, they're basically at the equivalent rate as what you see in low temperature organisms. And they also have to decrease the fluidity of their membranes. It's basically the concept you can imagine is, you know, butter versus margarine versus olive oil versus other things. The thermophiles need to pick the composition of their membrane such that at high temperatures it's got the same fluidity as the membranes of mesophiles at low temperatures. And so they play with things like cholesterol in their membranes to make it stickier. This cartoon I like because it's important to realize that we call a lot of these organisms extremophiles and we compare them to things like us. But that doesn't really mean that we've got the adaptation in the right direction always. The ancestral state for many of these organisms might be extremophily. And mesophily, like growth at low temperatures, might be the derived state. We don't know unless we map all of this onto a tree and figure out what are the ancestral and derived states. So it's important to think extremophile doesn't mean it's a new invention, per se. So again, there are lots of industries that want to use enzymes from thermophiles because there are lots of reactions that need to happen at high temperatures. And I have a list of these. I'm not going to go into all the details of them. But what we're going to talk about in the Friday lecture is this approach to studying microbes in the environment where we look at their DNA. And we're going to talk about how that applies to the microbes that live in and on us.